So this week, you sent over some really interesting research on how scientists are figuring out ways to boost production of like valuable compounds in plant cells that are grown in labs.
Yeah.
And it all comes down to this technique called elicitation.
Yeah, exactly.
So I guess to kick things off, could you explain what elicitation is and why this is such a big deal?
Well, in simple terms, you can kind of think of it as giving plant cells a little nudge to produce more of the good stuff,
Yeah.
right? The compounds that we're interested in.
Okay.
So elicitation is basically the process of applying these specific signals, or we call them elicitors, to plant cell cultures.
Mm-hmm.
And it's kind of like introducing a controlled stress.
Okay.
It triggers the plant's natural defenses,
That's right.
and in doing so, it often supercharges the production of certain metabolites.
I see. So it's like we're tricking the plant to thinking it's under attack, and that somehow leads to it producing more of what we want. So what kinds of signals are we talking about here? What exactly are these elicitors that get the plant cells all fired up?
So broadly speaking, we can group elicitors into two main categories,
Okay,
biotic and abiotic.
gotcha. So biotic elicitors are like biological in nature, right? And then abiotic could be the non-biological ones.
Yeah, you got it.
Okay, so let's start with the biotic ones. What kind of biological triggers can set off this whole process?
Well, a lot of times these biotic elicitors mimic what happens when a plant's being attacked, like in nature.
Oh, interesting.
So we're talking about things like microbial elicitors,
Yeah.
which are essentially substances that come from, microorganisms like fungi or bacteria.
Great.
For example, chitin to chitosan,
Mm-hmm.
which are major components of fungal cell walls. These can act as really potent signals.
So it's as if the plant cell is sensing a potential fungal invasion, and then it starts ramping up its defenses.
Exactly. It's like a little red alert goes off inside the cell.
And part of that defense mechanism is producing more of these valuable compounds. Wow, so it's like turning the plant's own immune system into a factory.
In a way, yeah.
Yeah.
And we also see this with certain proteins and even extracts from things like yeast.
Hmm.
Now, what's really cool about microbial elicitors is that we might be able to use them in a more targeted way,
Okay.
to enhance the production of a very specific compound.
Oh, so like tailoring the trigger to get the desired outcome.
Exactly.
Fascinating. Okay, so we've talked about these microbial elicitors that basically mimic a threat to the plant.
Right.
But what about plant-derived elicitors?
Yeah.
Are plants themselves capable of sending out these kind of alarm signals?
Oh, absolutely.
Yeah.
So when a plant gets stressed or damaged, it actually releases its own internal signaling molecules.
Okay.
And these compounds, they're called endogenous compounds, can also act as elicitors when we're working with plant cell cultures.
Wow, so it's like the plant has its own internal communication system,
Yeah, you could say that.
and we can tap into that to manipulate production. Amazing.
Ah.
Okay, so let's shift gears now and talk about the abiotic elicitors, the non-biological triggers.
Sure.
What sorts of non-living things can have this effect on plant cells?
Well, this is where things get really diverse. You see, abiotic elicitors can be physical stresses like UV radiation
Uh-huh.
or osmotic stress,
Mhm.
which happens when there's high salt or sugar concentrations.
I see.
And even temperature swings can act as elicitors.
Wow.
Oh, and we can't forget about mechanical stresses, like
Oh.
the kind of agitation that happens in a bioreactor,
Oh, right.
That could also trigger a response. And here's a surprising one. Even tiny amounts of heavy metals like copper can sometimes do the trick.
Really? So even things that we normally think of as toxic can be beneficial in the right amounts?
Yeah, exactly. It's
Yeah.
all about finding that sweet spot,
Right, that Goldilocks zone where the stress is just right.
Exactly.
Not too much, not too little.
So it's a delicate balance to strike.
It definitely is.
Okay, so we've got this whole array of physical factors that can influence these plant cells.
Right. But what about chemical elicitors? Do they fall under this abiotic category?
Yeah, they do. So these are basically non-biological chemical compounds that can trigger a response.
Okay.
And some of the most important ones are actually plant hormones, like jasmonic acid or JA and salicylic acid or SA.
Mhm.
These are key players in a plant's defense system. Then you've got things like inorganic salts, for example, vanadyl sulfate,
Okay.
and even things like ethanol or certain detergents can act as elicitors. But again, it's all about using very low, carefully controlled concentrations.
Got it. So we're essentially throwing all these different signals at these plant cells, whether they're biological or non-biological.
Right.
But how does this actually translate to more production of the compounds we're after? What's happening inside the cell?
Well, think of the elicitor as a key that unlocks a whole cascade of events inside the cell. You see, plant cells have these special receptors that recognize elicitors.
Okay.
And when an elicitor binds to its receptor, it sets off a chain reaction, a signaling cascade that involves all sorts of things, like reactive oxygen species and calcium ions.
Wow, it sounds complicated.
It is. But the end result is that this cascade activates specific genes inside the plant cell. And these genes are the blueprints for the enzymes that actually drive the production of those valuable secondary metabolites.
So it's like we're flipping a switch that turns on the cell's machinery to produce what we want.
That's a good analogy.
So can you give me an example of like a specific enzyme and the compound it helps produce?
Sure. So enzymes like PL and CHS are crucial for making phenolic compounds. And these phenolic compounds have all kinds of applications in medicine, cosmetics, and even agriculture.
Wow, so we're talking about some pretty important stuff here.
Yeah, for sure.
Okay, so I'm getting a clearer picture of how this all works on a cellular level.
Good.
But how do scientists actually use elicitation in practice? Like, when do they add these elicitors to the plant cultures?
Well, the timing is really important. Usually the elicitor is added to the culture medium during a specific growth phase, often in the light exponential or stationary phase.
Okay.
When the cells are really active.
Right.
But it's not just about when you add it.
Right.
You also have to carefully optimize the type of elicitor, the concentration, and how long it's exposed to the cells.
So there's a lot of fine-tuning involved.
Absolutely.
Yeah.
Each plant culture and each target compound is different.
Yeah.
So you have to figure out the perfect recipe for each one.
And the goal is to get the maximum yield without harming the cells, right?
Exactly. It's that delicate balance again.
Right. Not too much stress, not too little.
Precisely.
Well, it sounds like a really powerful technique. When it's done right, it seems like elicitation could be a game changer for producing these plant-derived compounds in a controlled way.
I think so. It's definitely a more sustainable and controlled approach compared to traditional methods of extraction.
Okay, so let me just make sure I've got this right. We're basically introducing these stress signals, these elicitors, to kind of trick plant cells into producing more of the compounds we're interested in.
That's the gist of it.
And it all comes down to understanding and manipulating the plant's own natural defense mechanism.
Exactly. It shows you just how responsive plants are to their environment.
It really does. It's amazing. And it makes you wonder, if we can use these techniques to enhance production in plant cells, what other biological systems might be out there just waiting to be optimized?
That's a great question, and I think it's a really exciting area for future research.
Absolutely. Well, thanks for taking the time to break all this down for me. It's been a fascinating deep dive into the world of plant cell elicitation.
You're welcome. I'm glad you found it interesting.
I definitely did. And I'm sure our listeners will too.
I hope so.
So to our listeners out there, keep those questions coming. And we'll see you next time for another deep dive.
See you then.